Hello students, today we will learn the Invitro Haploid culture, the second part of our present topic. This is the continuation of haploid culture. Today we will learn the process of anther and pollen culture, its advantage and disadvantage and applications. Uh, Now, what is an anther and what is a pollen? Basically, a flower, which is the reproductive organ of a plant, contains four different parts. Those are two protecting parts, named as sepal and petals, and two reproductive parts, named as anther and ovary. The anther and pollen are the part of the reproductive organ androecium, and the ovary is the reproductive part of the reproductive part of the organ known as gynoecium. The anther is the part of the stamen containing pollen. So pollen is present inside the anther. It is the fertilizing powder which is discharged from the anthers. So let's see. This is the total flower, and on the upper side of the flower we can see the anthers. These are anthers, and inside the anther the pollen is present. This is the filament or stalk on which the anther is located. This is the stigma. This is the stigma at the tip of the gynoecium, and this is the style. And after style is attached with the ovary, the female reproductive organ. The anther and pollen are the male reproductive organ here. So this is the anther, the yellow color. This is the filament, and it is the cross section of the anther, and we can see the pollen grains inside the pollen sac. So what is anther culture? Anther culture is the culturing of anther to obtain the haploid plantlets from it. It is obtained from the unopened flower bud in the nutrient medium under aseptic condition. The callus tissue or embryos from anther will generated from in this culture, and the haploid plantlets from this can be raised either through organogenesis or embryogenesis. And what is pollen culture? Pollen is the microspore that is cultured in vitro condition by obtaining the pollens from the anther, and preferably it must stay at its uninucleated stage, and that uninucleated pollens can be cultured to obtain the haploid through either embryogenesis or organogenesis. The principle of anther and pollen culture is the production of haploid plants. It is to exploit the totipotency of microspore. What is totipotency? Totipotency is the power of a cell to this can be uh, give rise. This can give rise to a whole organism. Basically, in plant cell, all cells are theoretically totipotent, but in practical, we can see that only the meristems and some soft tissues have the ability to generate a new whole plant, and those plant cells are called totipotent cell. In case of human, only the fertilized embryo is a totipotent cell. There are several potency level of a cell called totipotency, pluripotency, multipotency, unipotency, etc. Um, so, in case of plants, theoretically all cells are totipotent in nature. And in the process of normal development and function of the pollen cells is to become a male gametophyte is stopped and is diverted forcefully to new metabolic pathway for vegetative cell division. This is the main principle hidden under the haploid tissue culture principle. So, what is the method of anther culture? First, we have to collect the flower buds. We have to collect the mature flower buds, and these flower buds are surface sterilized with 70% ethanol, along with some other surfactant, other sterilizing materials known as. We can use the mercury chloride also, and after that the anthers are excised from the flower bed and kept separately. The anther is in the first meiotic division is selected by acetocarmine test. It is the test for cytology, where we can easily count the chromosomes, and we can easily identify the meiotic cell division with this 10. Generally we use acetocarmine for the meiotic cell division testing, and we use the acetoorsin solution or acetoorsin stain for the mitotic cell division. After inoculation in the medium containing some essential amino acid known as we can use glutamine or L-serine or inositol. The incubation is incubation period will incubation period will start, and after 15 days culture, the anthers can grow into embryos. The embryos are transferred to the rooting medium, and they kept in 33,000 lux illumination, and 4 to 5 weeks after that the embryos become plantlets. The plantlets then can be transferred for acclimatization, and they are transferred to the greenhouse. So by this process of anther culture we can easily get the haploid plants. So let's see this figure. This is the anther culture in haploid to generate a haploid plants, and anther is the onset of culture here, and B is the anther after 6 days culture. These are embryos emerging from the anthers after 30 days, and this is the shoot and root generated from the anthers, and this is the plantlet with cotyledons and leaves. These are the leaves, and this is the subculturing of the plantlets into growing medium, and this is the 80 days old regenerated haploid plants from anther culture. What is the method of pollen culture? This is basically the same, but we have to isolate the pollen first, and for this purpose we have to squeeze the anther, and from where the pollen comes out in a sterile. This all should be done in the aseptic condition inside the laminar airflow machine, and after that after obtaining the pollens we must obtain this anther in the liquid niche and white media, or we can also use the ms media, but it should be the liquid media. The anthers are then pressed against in the pressed against the side of the beaker to squeeze to obtain the pollen. The homogenized anther are then filtered to through the nylon sieve to remove the anther tissue or debris which are often deployed in nature, and we are getting the sterilized pollen, filtered pollen in the liquid medium. The filtrate of pollen solution is then centrifuged at low speed, and the supernatant containing fine debris are discarded, and after that the pellet of pollen is suspended in the fresh liquid media and washed after that, and it is resuspended in fresh liquid medium. In to culture in a 5 centimeter petri dish, we need at least 2.5 ml of pollen suspension, which generally containing 10 to the power 3 to 10 to the power 4 pollens per ml. and these pollen are best grown in liquid media because the osmoticum present in the liquid media can help the pollen to grow faster, and also we can grow it in the um, solid agar media, depending on the nature of the pollen. The petris are incubated in 27 to 30 degrees centigrade and low intensity of white light. The young embryos can be observed after 30 days, and these embryos ultimately give rise to the haploid plant. So after getting the embryos, we can subculture it in the rooting media and shooting media, and then we can culture it um, in the regeneration media of to get the haploid plants. So this is the basically technique shown pictorially here, the method of pollen culture. So what are the advantages of anther and pollen culture? It is with the advantages are with only one set of chromosome. This is a mutations are easily be identified. The doubling of the chromosome number with some treatment, chemical treatment, we can easily double the chromosome numbers, and we can easily produce a double haploid plant, which ultimately result in a completely homozygous plant. The use of double haploids in breeding program can greatly reduce the time required for the development of improved cultivars. Sometimes we also can achieve the gametoclonal variation. It is maybe useful in some cases, while often negative in nature, but some useful traits can be achieved with the gametoclonal variation through the haploid culture. Uh, in tobacco, the gametoclonal variation has been associated with an increase in amount of nuclear DNA, and this is very much helpful for the vigor generation. And it is simple and less time consuming and responsive. This is the main advantage. The problems associated with the anther and pollen culture, those are the disadvantages. The problems encountered in plants during or as a result of anther and pollen culture range from very low yields or is genetically unstable. Those plants are genetically unstable. So we have to make it double haploid to give it a genetic stability. So these genetically unstable plants can be genetically stable by means of by the help of double haploid production. In many of the major horticultural and agronomic crops, they do not yield sufficient haploids to allow them to be useful in the breeding programs. That is another problem. And in other species, genetically instability has often been observed from plants recovered from the anther and microspore culture. So I have already mentioned this, but we can easily recover from this disadvantage through the double haploid generation. The gametoclonal variation can be observed. It have some beneficial effect also. In case of tobacco, we also seen that we also saw that the beneficial effect in in many cereals. The high percentage of the plants regenerated from the anther and pollen culture are albino, and these albino plants cannot photosynthesis, and that's why the cytoplasmic DNA have been associated, and that's why they are not growing in the media, culture media, and we observed that cytoplasmic DNA have been associated with the albinism. It requires a skillful techniques. It is a skillful techniques, and that's why it's required a very skilled person to culture anther and pollen. And it is not much successful in case of cereals. Only in diploid plants it is successful. The applications are for mutation study. We can use the haploid plants. We can use these plants for breeding and crop improvement purpose. These are all highly in use to obtain the secondary metabolites. As for example, that H. niger is obtained by the anther culture having higher alkaloid contents. The haploids are used in molecular biology and genetic engineering purpose. In haploid tissue culture, Arabidopsis and Lycopersicon, that is tomato, have been used for the transfer and expression of 3 genes from E. coli. Those are already proved. So this is the these are the applications. In the next class, we will discuss how to culture and female reproductive organ of a plant, that is ovary culture, and after that we will discuss the how we can achieve the double haploids from the haploid plantlets, and after that we will discuss the ploidy level and its applications. Thank you.